In this video, we'll focus on the structural correspondence between one gene and one polypeptide. When Beadle and Tatum proposed their model of one gene and one protein, the relationship between a protein's structure and its activity was not well understood. We'll explore experiments that demonstrated the relationship between a mutation and the activity of a protein. The primary structure of a protein is defined as the sequential order of amino acids in a polypeptide. The secondary structure is the structural arrangement of adjacent amino acids in a polypeptide chain. These stretches of amino acids form stable secondary structures such as alpha helices or beta strands, denoted with the Greek letters. Usually, secondary structures are connected to each other in a polypeptide chain by many unstructured loops of amino acids. The tertiary structure of a protein refers to the three-dimensional folding of the polypeptide chain. It is stabilized by either non-covalent interactions or covalent disulfide bridges. The tertiary structure creates structural and functional domains that are responsible, for example, for the specific activity of enzymes. The stability and the conformation of the tertiary structure depends on the primary structure. Any changes or mutations in the primary structure might affect either the stability or the activity of the protein. Quaternary structure exists in some proteins that are made of different subunits. Either identical or distinct folded polypeptides assemble to form this higher order structure. Usually, the assembly of the subunits into the quaternary structure is necessary for the full activity of the protein. So, what is the correlation between a genetic mutation and the structure and activity of an enzyme? Mutations in genes can have different effects, including the inhibition of protein synthesis, the alteration of the structure of a protein, which leads to a loss of activity, or more complex indirect effects, such as the activity of enzymes. Let's delve into two examples to illustrate these concepts. The first example involves a mutation that directly affects the stability of a protein and thus indirectly affects its activity. The enzyme in question is tyrosinase, which is involved in the synthesis of melanin. So, what do we know about enzymes? We know that the activity of an enzyme is temperature dependent. We also know that exposing these enzymes to suboptimal temperatures may result in a loss of enzyme activity. To demonstrate the effect of temperature on tyrosinase activity, the enzyme was first maintained at an optimal temperature and then exposed to a higher temperature of 50 degrees Celsius for 0, 30, or 60 minutes. The consequences of this exposure to suboptimal temperatures on enzyme activity were then measured. When exposed to suboptimal temperatures for various lengths of time, the wild-type enzyme activity decreased. The enzyme containing the mutation, however, exhibited a much more rapid decrease in activity compared to the wild type. The data revealed that without exposure to high temperatures, there was no discernible difference in activity between the mutant tyrosinase and the wild type at 25 degrees Celsius. But when incubated at 50 degrees Celsius for 30 minutes, the wild type enzyme retained approximately 2/3 of its activity, while the mutant enzyme showed a complete loss of activity. The conclusion of this experiment indicates that the mutation alone does not render tyrosinase inactive. This is evidenced by the fact that maintaining the mutant enzyme at its optimal temperature allows for normal activity. Instead, the mutation affects the tyrosinase structure, making it less thermostable. Let's look at a different example. Consider hemoglobin, or HB, in humans. Hemoglobin is a protein in red blood cells that is responsible for delivering oxygen to the tissues. Some individuals suffer from a disease called sickle cell anemia, where oxygen is not carried efficiently in the body by these cells. Let's compare the properties of hemoglobin between healthy individuals and sickle cell patients. The isoelectric point is the pH of the environment at which a protein has a net charge of 0. If we calculate the isoelectric point for oxygenated hemoglobin from both a healthy individual and a sickle cell anemia patient, we can observe a difference. the sickle cell and normal hemoglobins differ by 2 to 4 positive charges. The difference in net charge suggests that in the hemoglobin of a sickle cell patient, a mutation that results in an amino acid substitution has taken place. To test this, an experiment can be conducted. We can take hemoglobin from both a healthy individual and a sickle cell anemia patient, and then subject this purified hemoglobin to trypsin. Trypsin is an enzyme that cleaves proteins specifically after arginine and lysine in the polypeptide chain. This creates fragments of hemoglobin called tryptic fragments, which are produced from the digestion with trypsin. These fragments can be obtained from both healthy individuals and patients with sickle cell anemia. To compare these tryptic fragments, a two-dimensional separation is performed. In the first dimension, electrophoresis is conducted, which separates the tryptic fragments based on their net charge. In the second dimension, chromatography is performed, which separates the fragments based on their hydrophobicity. The expectation is that each tryptic fragment will end up in a specific spot in the final chromatogram. The position of these spots depends on both the net charge and hydrophobicity of the tryptic fragment. The experiment starts with a mixture of tryptic peptides that are applied at the corner, followed by electrophoresis in the first dimension. This results in separation based on charge. Here, only 4 spots are represented, but under normal conditions, 28 spots would be obtained. Among the 4 spots, spot 3 shows a different electrophoretic mobility in the hemoglobin A from a healthy individual compared to the hemoglobin S found in patients suffering from sickle cell anemia. This difference is further observed in the second dimension through chromatography, where the peptide hydrophobicity determines migration. The position of spot 3 is different between hemoglobin A and hemoglobin S. The interpretation of this experimental data therefore involves focusing on the amino acid sequence of tryptic fragment 3. A trypsin treatment produces 4 fragments, and upon separation using both electrophoresis and chromatography, a difference in fragment 3 is identified. The difference in charge and hydrophobicity suggests a difference in composition. In hemoglobin A, a glutamine, or GLU residue is present, while in hemoglobin S, it is replaced by valine, or VAL. This amino acid substitution in hemoglobin S changes the electrophoretic mobility, chromatographic mobility, and hydrophobicity of the whole peptide. This example illustrates how, given an analysis of a protein structure, in this case hemoglobin with a mutation that is associated with sickle cell anemia, revealed a correspondence between the mutation and a change in amino acid. Glutamine, a hydrophilic and negatively charged amino acid, is replaced by valine, a hydrophobic and neutral amino acid, thereby changing the composition and activity of the peptide. This change in amino acid is responsible for the altered structure of hemoglobin, which causes it to aggregate and change the shape of red blood cells from their normal biconcave shape into a sickle shape. This structural change is the main cause of sickle cell anemia and explains the associated symptoms. In conclusion, these experiments help to refine our definition of a gene. A gene is a unit that contains the information necessary for the production of a biologically active polypeptide chain. It is also a unit of function that defines the primary structure of a polypeptide chain. Mutations in a gene can lead to changes in the primary structure of a peptide chain, which affects the activity or stability of the protein.